Today I'm going to be talking to you about extractables and leachables and how a study we performed on a single-use bioprocess system where we're going to examine its extractables and leachables. But really the goal of my talk is to give an overview of how this process is conducted and some of the unique ways that we do this at Jordi Labs. By way of introduction, I just wanted to tell you that Jordi Labs is a company that was founded in 1980 to analyze plastics. We do over 1,000 analyses annually. And those analyses range from things like extractables, leachables, to things like investigations of failures, cracking, off-odor problems, a wide range of polymer analysis issues that will help our clients to tackle. So in my talk today, I'm going to cover, first off, E&L background. What is an E and what is an L, and how do you conduct this type of testing? I'm also then going to go through a model study in which we're going to look at a bioprocess system and look at the process and what we found as we actually did an E&L study on this. So first off, what is a leachable and what is an extractable? A leachable is a component which extracts out of a material under the intended use conditions, whereas an extractable is a component that comes out under an exaggerated condition. So really it's the condition under which it comes out that determines whether it's a leachable or an extractable. And in general, the types of things that we find when we start doing an E&L study is we'll find things like antioxidants, slip agents, lubricants, many of the different types of things that are contained in a polymer system that are small molecules. But some of the most interesting components that we'll find when we do an E&L study are those which are either byproducts or oligomeric materials of the polymer itself. And those present unique analytical challenges because of the fact that many of them are not commercially available. So then the question becomes, well, how do I quant something if there's no standard? How do I identify something if there's no standard? And so those are really where some of the challenges come in E&L work. The regulatory landscape that surrounds E&L testing is broad. There's different regulations for each market segment. So in terms of pharmaceutical packaging, they have their own regulations, and as well as bioprocess systems. And then also, if you're doing medical devices, they want to follow typically ISO 10993 and have their own FDA guidance. And lastly, there's unique E&L regulations for food contact applications. So you really have to look at each project individually and ask the question, what type of sample am I analyzing? What's its intended use? In order to know what regulatory landscape you ought to follow when doing this type of testing. So this slide is an overview of what an E&L study consists of. And the first section of an E&L study really is gathering background information. In order to design an E&L study, you have to know a lot about the device that you're going to study in terms of what is it made of and how is it going to be used. So those are the two primary questions we need to answer. In addition, there's other things we need to know, like what is the surface area of the device, but it's important to have a lot of background information before you design your study. After we get through that phase, the next step is to go through and devise a protocol for how the testing is going to be conducted. And typically, in the protocol, we're going to detail what samples we're going to analyze, what controls, what analytical methods we're going to use, and also how are we going to conduct the extractions. After that phase is completed, we then go into the chemical analysis phase where we actually perform the testing itself. And lastly, you're going to do a toxicological evaluation of what you found. So once you've got your list of here's what extracts, and here's how much of it there is, you then need to do toxicology in order to answer the question, is that relevant? Is it toxicologically significant? So that's the last phase. So as I mentioned, I'm going to walk you through this process in terms of a model study that we did on a bioprocess system. And by way of introduction, this system consisted of three main components. It had a bioprocess bag. It also had a tubing that's made of a thermoplastic elastomer, and then lastly there was a filter that was used, a filter system. So it's those three components that make up our bioprocess system. I think my battery may be going, but... So in terms of the study design for this particular analysis, we did several different kinds of extractions. The first is what's called cut and cover. In cut and cover, you basically section the sample and then completely submerge it in the extracting fluid. In addition to that, we also did some flow-through extractions where we actually replicated the use condition and flowed continuously the solvent through the system to try and replicate the condition under which it would actually be used. These extractions were either conducted at 50C for 72 hours or 37C, the 50C being an exaggerated condition. So anything that we find under those conditions would be considered extractables. The 37C was done for our leachables condition to be similar to what you would expect the use condition to be. And we did a range of different solvents. Typically, you want to do solvents of different polarity. So we used saline, we used water, we used 50-50 ethanol, water, and we used 100% ethanol as well. And then we also did that on each one of the components individually, as well as on the system as a whole, so that we could compare the extractables that came from each portion of the device. Now, once we had our extracts, the next thing we need to do is to start analyzing them. And so we chose to use a series of screening methods or scouting methods to do initial studies. And the purpose of your scouting methods is really to get a good idea overall what's in there. You're not trying to identify individual extractables and leachables. You're trying to just say, in the bulk, do I have a lot of extractables? And if so, what are their general properties? So we used FTIR to get a general sense of the bulk chemistry. We used gravimetric analysis to tell what the mass was of the total extractables. We used TOC to look at the total organic carbon. And we used UV-VIS to get an idea if there were chromophore or aromatic containing species that came out of the material. After we do our scouting methods, we're then going to do a series of more definitive analytical techniques, things that are designed to look for compounds of various volatilities. So we did 4 different techniques in this study. We did dynamic headspace, which looks for very volatile compounds. And then we did GCMS, which is for semi-volatiles. We did LCMS, which is for our non-volatile and semi-volatile compounds. And lastly, we did ICP-MS to look for elemental impurities. So those are the 4 tests we're going to use to identify individual leachables and extractables. Now, once you've chosen which techniques you want to use, another question you have to answer is, how sensitive do I need my methods to be? If you're doing an extraction and you use more solvent, you will in effect dilute your extract, and you will then have less of that compound per mil of solvent. So you need to set from the beginning a threshold of how sensitive do I want my methods to be. And that threshold is set using the concept of the AET, or Analytical Evaluation Threshold. And basically the AET just defines what is that toxologically relevant level above which I need to identify a component if it's present. One thing I wanted to mention about the AET is in the current regulatory landscape, this concept is only defined for pharmaceutical packaging. So there are no definitive regulations as regards medical devices or bioprocess systems yet. And so we have to adapt those pharmaceutical regulations to apply them to medical devices and also to bioprocess systems. So as I mentioned, the AET determines what level is toxologically relevant. So if we, for instance, set that AET value at 15 nanograms per mil, then only the components that are above that line would need to be identified. This is an LCMS chromatogram, and so we're saying only those peaks that are higher than that have a larger peak area than that, would actually need to be identified in our study. But if, on the other hand, we were to set that level lower and set that at 25 nanograms per mil, then you can see we'd have to identify a lot more components. So that's, again, a very important decision that's made during the study, is how low is low enough from a toxicological perspective. Now, in order to determine where that line is on a chromatogram, you have to run a standard. So one other important decision we have to make is which standard are we going to run to set that intensity threshold at. And if I, in this case, chose arusamid as my standard, then my line for that 25 nanogram per mil would be where you see it right now. But if I chose a different standard, I'm going to get a different line that's related to the signal response that I get for that particular standard. So one of the very crucial decisions that has to be made is, what is the right standard to run in any of these kinds of studies? And in our experience, that's something that's not given enough consideration when people are choosing the conditions they're going to use to run a study. So we have looked at that in depth, and this slide just summarizes the relative response factors for 14 different polymer components that are commonly used. These are slip agents, there's some plasticizers in here, and antioxidants. And what I want you to get from this slide is the fact that any compound that has a relative response factor below 100 would be excluded from your study if it was at the AET value, even though it's actually at the toxicologically relevant level. So many times, again, people don't think about that, but when we think about safety, and we think about the safety of a bioprocess system or a medical device, if we don't choose the right standards, we can exclude compounds that are actually present at that toxicologically relevant level. So how do we handle that at Jordi Labs? Well, we've designed a very unique approach in which we are using a triple detection system. This system has UV-VIS detectors, it has a CAD detector, and it also has a MS detector. And with all three of those detectors, we can then choose the detector that has the most universal response for any given class of compounds. So what you see here is for the first class, which are slip agents, that's starting with a ruselamide and a leamide, those have a more generally universal response by MS than by the other detectors. If, on the other hand, we're looking at the phthalates, we're going to want to go with the UV-VIS detector, which has the most universal response. And then if we're looking at the class that's the antioxidants, typically either the UV or the CAD is preferred depending on the volatility of that particular antioxidant. So this slide just shows how if you calculate the RSD for those various detectors, which one would give you the best and the lowest variation in terms of overall response factor. And again, you can see that those trends line up where we want to carefully choose that detector when we're doing this work. So one of the things that I think really is crucial in this overall process, again, is A, to make sure you pick the right standard, because if you don't pick the right standard, things can be excluded from the study even though they're toxicologically relevant, and B, to pick the right detector so that you can make sure when you're doing this work, you're not getting more error than you would otherwise need to. All right, so now that we have set up our study, we've chosen what instruments we're going to run, we've chosen what methods we're going to use, and we've chosen what level of sensitivity we need. And if I could get my slide to turn. I apologize, guys. Could you advance the slide, please? Thank you. There we go. Thank you. So now that we have chosen the conditions we're going to use for our study, the next step is to actually perform the study. And we can advance the slide again, please. So this is the first slide of the actual results of our analysis of this bioprocess system. And these are the gravimetric and TOC results. And what you'll see here is that generally in our extractions, we saw low milligram quantities of extractables in this particular case. We did have one exception, though. If you look at the tubing sample in the ethanol, we saw almost 2.5 grams of material that came out. One of the questions we then asked is, did the material degrade under this condition? And the answer is no, it actually was still intact. We just know that a very large amount of material came out in that particular sample under this extraction condition. When we then examined that with FTIR, we can see that that material is consistent with mineral oil. So that particular tubing had a mineral oil in it, and that's what was coming out in such large quantity. And that's one of the reasons we like to do these scouting methods, is just to get a general sense of what's coming out. We then did the UV-VIS analysis, and we could see that most of the extracts contained either chromophores or aromatic species, because you could see absorption up to 290 nanometers. Go back one more, please. Thank you. And then now we began to do our individual, more sensitive methods to look for individual extractables and leachables. And this is an example of the dynamic headspace results. And this is a method that often is overlooked when people are doing E&L studies. They only tend to do LCMS, GCMS, and ICP-MS. But in our laboratories, we like to use DHS as well, because some components are so volatile that if you try to extract them in a solvent first and then analyze them, they will be lost, either because they'll be buried underneath the peak in the GCMS, or because when you try to concentrate them, you will lose them. So dynamic headspace is very good for that. And this is an example of where we're seeing some very volatile components, ethyl acetate and some acetic acid that would easily be lost if we were doing just conventional GCMS. And we then did ICP-MS on all the extracts, and you can see that we generally got very low quantities of elemental impurities, nanograms per device, so all of these were below the accepted toxicological thresholds. We did see a little bit of tin in here. And if we could go back one slide, thank you. So one thing I wanted to mention, one of the unique features that we like to use in this particular instrument is we can analyze over 11 orders of magnitude of linear dynamic range for this instrument. So we can analyze all of these low nanogram per device levels, while at the same time having a salt background of 350,000 nanograms per mil. So that's very useful for looking at saline extracts. This is an example of the GCMS chromatograms that we obtained. And you can see there's a large number of peaks here, but there is one component which is consistently seen throughout a lot of the extracts. We can advance one more, please. And then this is an example of the LCMS chromatograms that we observed. Now, when you start looking at GCMS and LCMS data, many laboratories will typically go through this, and it's effectively a manual process or a semi-automated process in which they will click on each of these peaks to identify what components are present. In our laboratory, we don't use that process. I want to take you through a little bit of what we do use, if we could advance. So when you start looking at one of these GCMS or LCMS chromatograms, the question becomes how many components are actually present in this particular chromatogram. And if you're looking at it zoomed out, you might say, well, maybe there's 50 components. But if you start zooming in, you can see, well, actually, there's a lot more components present. And actually, if we could zoom in one more time, we'd see there's even more components present. And any human being trying to go through and figure all of this out on their own is not going to be successful. So we have implemented a different approach, if you would like to go forward. We use a combination of software packages which mine this data for unique mass features at each retention time. And then we're going to use statistical analysis to do differential analysis on a sample versus a control to ask the question, what is actually unique in this sample that's truly an extractable, and what's a background component that's due to just the solvents that you use to do the extraction with or the glassware. So to take you through that process just a little bit, the first step of that process is called recursive feature finding, and we'll go ahead and advance, please. The recursive feature finding step basically is going through the entire chromatogram, and at each retention time we're asking the question, is there anything that is unique in terms of mass at this retention time versus all the other retention times? And so you can advance. When we do that process for our extracts, we found this many unique mass features. And so for each of the extracts, if we take, for instance, the ethanol extract of the tubing, you can see that we found 922 unique mass features that were present in that particular chromatogram. So that's quite a few mass features. So the next step we're going to do is we're going to do statistical analysis to ask the question, are those features unique to the sample extract as compared to a control extract? We're also going to apply what's called a fold change filter. And the fold change filter is going to ask the question, is the amount that's present in the sample greater than the amount that's present in the control? And when we do that, we can take the extract that had 726 compounds, and 288 of those are explainable based on the control. That leaves us with an additional 575 unique mass features that we would now still need to identify. So we're going to apply yet another filter. The filter we're now going to apply is that AET filter we were talking about earlier, and this is obviously a very important decision because we don't want to miss things that are actually at a toxicologically relevant level. But this filter, as I mentioned, is done by calculating the AET value. So in our study, we're going to make a couple assumptions for the purposes of the demonstration, and we're going to say that our Bioprocess system had contained 500 doses of a medication, and that a patient was to take 2 doses per day. If I use that and apply an SCT value of 0.15 micrograms per day, I'll calculate my safety concern, or my level, my AET level at 37.5 micrograms per device. Now, if I apply that level onto the chromatogram, you can see that there aren't very many components that I would need to identify. But as I mentioned earlier, if I don't pick the standard with a matching ionization efficiency to the compound under study, I'm actually underestimating how many components there are that are toxicologically relevant. So PQRI has recommended that one way to deal with that is to apply an additional safety factor. And that safety factor is either 50% or the percent RSD of your response factor database. And in this case, that's 83%, because as I showed you that list of compounds we analyzed earlier, there's quite a bit of variability there in response factor. That gives us a new revised AET of 6.4 micrograms per device. And if we apply that level onto the chromatogram, you can see then there's quite a few additional species that could very well be toxicologically relevant that we're going to choose to identify. So now when I've applied this filter, you can see that I've gone from 575 down to 202 components that are actually at what is believed to be a toxologically relevant level for this particular extract. So the last step of my process then is to identify those 202 components. And so I'm going to just show you how we do that. So to identify those components, I use a combination of the following things. First off, we have what I believe is the largest database of extractables and leachables in the industry. We've been building that database for over 30 years. And so that is a huge portion of how we do the identification. We'll search all those unique mass features against our proprietary database and come up with hits for those. We also have commercially available libraries that we'll use, like the Agilent E&L database, as well as we will use what's called molecular formula generation, which is an algorithm that calculates the best molecular formula to match the mass that we observed. And then after we've done that basic level of identification, we'll go another step and we'll look at the MS-MS fragmentation patterns as well as looking at analysis of standards to confirm identifications. And after we got all done with that, we came up with a list of the components that were present, and that particularly big peak that we saw in the GCMS was caprolactam, if you're interested. But we found a lot of things that make sense within a polymer system. So in summary, the process for the identification involves statistical analysis to figure out what is unique to your sample as compared to your control. And then we remove all of those components that are not unique. And then we go through and we identify each of those constituents that remain after we've applied those filters. So the last thing I wanted to show you is just some summaries of the data as a whole. This is all of the extractables from the tubing sample in both the water, the 50% ethanol, and the ethanol extract. And you'll notice that it's surprising how many are overlapping for various extracts. There's actually not as many as you might think, indicating that solvent polarity is a very important consideration when you look at what's going to extract out of a polymer system. In this case, there's only one extractable that happens to be present in all three extracts. And if you're like me and you're curious, you will see that that particular component is actually an ethoxylated compound, and those have a surfactant-like property, which is why it would come out in a wide range of solvents. And one other comparison is this one. This compares the saline versus all of the other extractable solutions for the tubing, and you'll see that there are 17 components which are unique to that saline extract. And the point I want to make with this slide is just to say that it's very important what condition is chosen for the leachables because every solvent has its own unique solvent strength, and even though we've done 3 other extractions with stronger solvents, there's still 17 unique constituents. So a lot of the work that's going on now is in how do we get a better leachable solution. For instance, if a device is going to be implanted and it's going to be in contact with, say, saliva or urine, then how do we get a better simulant than that than just saline? Because we've found differences in what we'll extract based on those differences that are present in those kinds of body fluids. All right? So with that, I will close, and thank you for attending, and thank you for your attention, and I'd be happy to take any questions.